Hello and welcome to today's webinar, and thank you for your patience. My name is Trevor. I'm the Communications Manager for the Canadian Cancer Survivor Network. Thank you, everyone, for attending today's webinar, the first in the fall leg of the 2024 season. If you're new to our webinar, a little background on our group. CCSN works to connect patients, survivors, and other stakeholder groups with decision makers and the wider community to engage in discussion and to act on evidence-based best practices to alleviate the medical, emotional, financial, and social costs of cancer. To learn more, visit survivornet.ca. This webinar is being recorded and will be available on YouTube, and a link will be provided in the email that you registered with, along with a link to the slides provided with this presentation. And we will add a quick Q&A session at the end, but if you'd like, put your questions in the chat function in the GoToWebinar tab. And now, without any further ado, I will quickly introduce our presenter. Don Husereau is adjunct professor of medicine at the University of Ottawa. He does freelance healthcare research and works with private and public sector life sciences organizations to help them understand the value of health technology and implications for health and innovation policy. And so, without any further ado, I will hand it over to Don for his talk, Harnessing the Power of T Cells.
And Trevor, can I confirm you can see my slides?
Yes, I can see your slides.
Thank you. And I do appreciate everyone's patience and apologize. I had a very strange computer problem, and I hope I'm begging forgiveness from this crowd. Thank you. And it is an honor to always be asked by the Canadian Cancer Survivor Network to do these. I'm going to give a kind of overview of harnessing the power of T cells and what you might be able to expect from this new therapies. My name is Don Husereau. I'm an adjunct professor at the University of Ottawa. I actually do work for both public and private sector companies, including life science companies who may develop CAR T therapies and bispecific T cell engager therapies, and also public and not-for-profit organizations that may be confronted with how to pay for them. I've worked at CADIS or CDA now in the past for 10 years. I've served on expert committees as a health economist. I may do so shortly. And I'm also involved in the health economics community. I'm interested in technology and technology policy. I'm hoping you will be able to understand by the end of this what these things are that we're talking about, sort out the hope from the hype, and maybe some of the potential benefits and harms, and maybe understand a bit about lived experiences with these new, what we call advanced therapies, and some of the current policy and care challenges. It's not going to be an intensive deep dive, and you're not going to be asked to explain some of these diagrams, and I can guarantee you, if you look for diagrams of CAR T and T cell, you'll find too many of them, and some of them are very complicated, like this one here. I thought I should explain what T cells are, just to begin with. The T is for thymus, because I think originally a lot of them were found there. They're actually found in the bone marrow, sorry about the spelling error, thymus, and lymph tissue, and in your bloodstream, so they're throughout your body. Whites, they are considered white blood cells, and they go into the thymus actually to kind of be trained for recognizing bad things in the body, including but not limited to cancer cells. And when we talk about T cells, and some of this might be familiar from the days of if people paid attention to HIV, people with HIV had a kind of T cell depletion because the virus was attacking mechanisms that produce these cells. And we talk about two different kinds of T cells. One is called a CD8, and the other one's called a CD4. The CD4 helps the CD8 to kill, and what it kills? Well, it kills cancer cells, among other things, and it does that by being trained to recognize and bind to things that are not part of the body, and then releasing all manner of chemicals to induce the death of that cell. And this diagram tries to illustrate what goes on. The CD4 comes in, it's secreting chemicals to help the CD8 do its job. The cell eventually dies, what's called apoptosis, it actually self-destructs, and then another cell comes up and cleans it up. And that other cell that comes up and cleans it up actually helps train other cells to recognize the cancer cell. It's a system that works very well, but unfortunately, evolution has not been favorable to us, because evolution wants us to do well, at least within the first 50 to 60 years of life. But as we get older, due to a variety of factors, these things kind of break down. T cells don't function as well, and that's where cancer can step in. So, as I mentioned, T cells can recognize and attack cancer cells. They can do some other things too, and ultimately they provide the memory and long-term immunity. I saw one diagram. I thought this was interesting on the internet, saying there are, kinds of cells, antibodies, and then cells like macrophages, neutrophils that are like first responders, but the T cells are kind of like the police headquarters. They go back, they remember, they interrogate, and then eventually jail, they kill cancer cells. So we've been studying these things for quite some time, decades, and we've started to learn how we can harness the power of them. And the first thing people may have heard of in the cancer sphere, and I apologize, there's another typo on here, the PD-1 inhibitors and the PD-L1 inhibitors, which is on the right, are types of drugs that came in and revolutionized the care of certain cancers. PD-1 is just the name of a protein that's on T cells, and PD-L1 is the name of a protein that's on B cells, which are activating these T cells. And so if we inhibit them, the PD-1 and the PD-L1 functions are a kind of break when they are activated. So you have to, it's kind of hard to figure out, but when the B cells activate the PD-1, it's a break. If we block that, we're basically blocking the action of the brake. We're putting a brick under your brake pedal, which makes the car go faster, which is a good thing if we're trying to kill cancer. And so these things are thought in colloquial terms to reinvigorate T cells that have not quite staved off cancer. And what we see is, in some cases, even cures for certain cancers that were incurable, like lung and skin cancer. So that was great, and we still see emerging PD-1, PD-L1 inhibitors, but scientists have decided, well, how come we can't take this a step further? And that's what the CAR T therapies are. CAR T stands for Chimeric Antigen Receptor T cell receptors. And what they're doing is, this is a very advanced therapy. They're removing T cells from your body, and these are T cells that would bind to cancer cells. They're removing them from your body. They are inserting a gene for this antigen receptor. This gene is intended to help the T cells better recognize cancer. Then they're growing up a bunch of T cells in a lab. Then they're reinfusing it into you. And lo and behold, the action starts to happen. The CAR T cells, now you have plentiful CAR T cells with all of these receptors for cancer, and they start to kill. And that actually speaks to how effective these agents can be. Also speaks to how harmful they can be. Everybody knows that when your body is fighting an infection or fighting a foreign invader, things can get a bit heated up. We have a bunch of CAR Ts in Canada already, including Chimeric, which is the first thing to come along, used in hematologic cancers like acute lymphoblastic leukemia, and B-cell lymphomas and follicular lymphomas. But now, since then, we've had some other incumbents. Absema came in 2021 for multiple myeloma. It was actually not positively recommended, I think probably due to maybe data not being quite ready for prime time. I can't quite remember the reasons. And then a few others. And you can see all of these cancers are actually hematologic, or these are all lymphomas or leukemias, hematologic in nature. And so there's a whole bunch of CAR T cells, and they're going to continue to come. And of course, like a lot of cancer therapies, They come in later lines of therapy. So, you know, patients who with a high unmet need, people who have failed the first line, the second line, the third line of therapy. Now let's bring in the CAR T, see if we can actually do something. But as these get more and more investigated, you're going to see them in earlier and earlier lines potentially of therapy. So that's the CAR T. The other is the bispecific T cell engager, and you're going to hear this term BiTE, which is bispecific T cell engager. BiTE is a bispecific antibody. So we have antibodies in our blood system. We produce antibodies naturally, but the antibodies we produce naturally, what are called IgG type antibodies, look like the one on the look like the red and purple thing on the right. They're Y-shaped little proteins, and the ones that we produce naturally typically just bind to one thing. So both of the little things at the top of the Y typically bind to one target. It could be a bacteria or whatever, a fungus, a cancer cell. But we've engineered these things to actually not just bind to one target, but to bind to two, and even we engineer things to bind to three now that are coming, dual binding. So these are engineered, one arm binding to one arm binding the other. So somebody got the bright idea, well, why don't we just have, why don't we make one of these things so that one arm binds to a T cell and the other arm binds to the tumor cell? Now that T cell has not been specially activated or trained, or there's no genetic alteration of it like with CAR T, but scientists knew that if you bring a T cell close to a tumor cell, just the proximity of it actually activates a whole bunch of things in our body that will potentially kill the T cell, or sorry, potentially kill the tumor cell. And so these little antibodies go in, they recruit T cells, they bring them close to cancer cells, and then the magic happens. That's the idea. And so this is not some, you know, the idea of an antibody or an antibody injection is not something new. We've been doing it, you know, for 20 to 30 years in other areas of other therapeutic areas. And there are already 3 bispecific T cell engagers. The first one actually was years ago, back when I was on committee, blinatumomab, a shocker to payers at the time because it was quite expensive. Now sometimes referred to as a first generation bispecific T cell engager. The ones now that we're talking about are these newer bispecific T cell engagers, Columvian Valley, the teclistamab, glofitamab, and they've come out more recently. So what are the differences between CAR Ts and BiTEs then for you? Well, or for somebody that wants to use them? Well, CAR T, I've already mentioned, is personalized. We're taking your own T cells and we're modifying them. Bispecific T cell engagers are just right out of a box. You just take it, reconstitute it, inject it. CAR Ts are typically just a one-time therapy because once we've introduced these T cells back into you, we assume that they'll sort of go on working, I guess, for some time. Whereas bispecifics, we have to continue to inject, kind of continue to recruit these T cells to kill tumor cells. CAR Ts, more complex manufacturing. Well, and it's not manufacturing in some manufacturer, not some factory out in Europe, but we're talking about manufacturing on the ground where we're taking cells out of your body and then having to get them back into. So you can imagine that that requires special infrastructure in order to do, whereas bispecifics are just simply, they can literally be manufactured in Europe, shipped over. Longer lasting CAR T, we think, and we think bispecifics may be more temporary. There's a question mark around that. Both are effective. And as I said, because we're activating and harnessing the power of T cells, it creates a lot of fury in your body. And so, and to the point where you need to be monitored. With CAR Ts, these things absolutely require monitoring. So you may be admitted to a hospital. With bispecifics, well, there's some question. I mean, certainly there's some idea that they do require monitoring, but as we go forward, we might see that the monitoring requirements might be not as severe. I think the big things that distinguish them, though, are this one-time versus repeated dose. That's probably an important thing to be mindful of. The side effects are a bit different, and maybe we get more durable responses out of the CAR Ts. And that's a debate. And as a matter of fact, I took this out of a journal of sort of a very highly regarded journal among hematologists, where they had a counterpoint. Are CAR Ts better than BiTEs, or are BiTEs better than CAR Ts? Well, in terms of effectiveness, that depends. It depends on what you mean by better. But basically, I think a lot of oncologists might say, well, we could give one or we could give the other, or it depends on the patient. Because side effects might be less severe with one, we might be tempted to give it to a patient who's more frail, et cetera. I'll get into that. What about administration? Well, I mentioned this manufacturing procedure. Collecting your T cells takes 2 to 3 hours. Modifying your T cells and then growing up in the lab takes several weeks. And that means that we actually have situations where you might qualify for a CAR T therapy. You may get your T cells collected, and then something can happen in the interim. You may have a medical event, and you may not be able to actually receive your therapy. BiTE therapies, well, we give them immediately. The reason we call blinatumomab a first generation, it was given by IV and it required sort of a lot of intensive treatment. The newer ones can be given every week to 2 weeks, and they're just given subcutaneously, just like the way people inject insulin. So not a drip, you're not as intensive a type of therapeutic regimen. And what about the harms? Well, there are harms that are common to both. And this is the thing that I was talking about. When you're starting to harness T cells, all hell breaks loose. You have this, what's called a systemic inflammatory response. Or what they're calling a cytokine release syndrome. Essentially, your body's immune system gets activated to the point, just like a very severe fever in a disease, where it could actually do you harm. And so they actually have to monitor, potentially give you medications to make sure that this cytokine release syndrome either doesn't happen, or if it does happen, you're going to be okay. Whereas, and there's another potential issue called an immune effector cell-associated neurotoxic syndrome, ICANS. So neurological toxicities, which are a bad thing to get, are also associated with both therapies. And right now, we think CAR T therapies may have a little more of the potential to create this severe neurotoxicity. Again, what does it mean for patients? It means they have to be monitored, maybe admitted to hospital. There certainly has to be a lot of people attending to them, and they may need therapies to kind of mitigate the risk of these toxicities creating problems in patients. There are differences, though, between the therapies in terms of side effects as well. CAR T therapies may result in more severe toxicities. BiTE toxicities might be milder. Of course, this all depends, but might be able to be managed a little more easily. BiTEs offer the advantage that if something really bad happens, I can just stop giving it to you. I don't have to give you next week's dose. CAR T, once you're in, you're in. And so we have to at least see what's going on. Now, these toxicities often happen toward the beginning of the therapy, or you know, if you're going to have it, you're going to have it right away, type of thing. I suspect the monitoring requirements sort of taper over time. Well, I don't suspect, I know. CAR T therapies, because they may have a little more harm, oncologists might be tempted to give them in people who are younger and healthier and who think can potentially tolerate a bad toxicity event, whereas BiTEs might actually open up the door for people who have morbidities or more frail, etc. What about lived experiences? What do patients think what have they reported with these things? Well, we know that patients are actually relatively satisfied with either therapy compared to alternative options. So when patient satisfaction has been measured, we do know also that increased anxiety has been reported about treatment outcomes, right? You're being given this thing that you're being told is potentially curative, but also could potentially create a really bad side effect. That's not something anybody really, that creates a lot of emotion. And there's been a note that there is a need for kind of emotional support mechanisms because everything's a bit of a gamble, and including the harms and the benefits. And then nobody really wants to be admitted to hospital or have a prolonged hospital stay. Obviously, intensive monitoring can be stressful. And obviously, if you're in that situation, you may feel very confined and isolated. It might not be the best experience. So, we already understand that patients have experiences with systemic chemotherapy and long stays on a chair, an IV drip, and here now we have something that's even more intense. So you can imagine it's something that a person has to be prepared for. I'm speeding along as well. So policy challenges with CAR Ts and these bites. Well, first of all, they're very expensive. And I just decided to kind of get out some numbers. This is from the CDA reports that have come out recently for both glofitamab, a BiTE, with an estimated 3-year budget impact of $3 million. Now, I have to be fair, this is for the same disease, but not necessarily for the same patient population. So it could be a bit of an apples to oranges comparison, but I think it gives you the point. Whereas Carvykti had a estimated budget impact of $340 million. So think about that in terms of expense, and that doesn't even include, these are the budget impact. This is the expense to the formulary manager, to the drug budget over 3 years in Canada. This doesn't even count all the nurses, all the doctors, hospital beds, and all the things that surround care that are tremendously expensive. But let's bear in mind that drugs and doctors are an important component of spend in Canada, but hospitals are even a more important spend, and there is a scarcity of beds and resources, and increasingly so. So this is this is as much of a finance problem as an implementation problem. And this slide kind of reflects what I'm just saying. You have to have these complex treatment protocols. You have to have people at the ready. You have to have a lot of coordination of human resources and infrastructure. And these things are often undervalued. You can write a check to buy more product, but you can't just write a check to free up more hospital bed space, or just put up a bed wherever you want. It's difficult to produce more of these things quickly, or highly specialized human resources. In some provinces, we know, and I've just spoke actually to one of the manufacturers of these products today, the compassionate use of these products, which was given, so sort of an only in research or a compassionate use, is difficult. So even if the physician says your only hope is this new therapy, even if a physician says your only hope is this new therapy, it's not funded yet, but the manufacturer's providing it to you free, they still might not be able to give it to you, because if you're in charge of the hospital and you have to create priorities about who gets beds and who gets access to human resources, you might prioritize, or you will prioritize, the things that you pay for versus the things that are still investigational or that other people are paying for. So this is a problem. If you're the patient, you want this. And if you're the oncologist, you want this. but they require all this other infrastructure. They're not available in the box, so to speak. Where, this is not a pill that you quietly go home and take. This requires people to look at you. So there are people investigating whether we might be able to take these bites at least on an outpatient basis, but healthcare administrators have a primary responsibility, if you look at anybody's website, for the safety of patients and to make sure that there's a quality of care. So decisions to let people go away with a product that could potentially end your life, not save it, are big decisions. And so some of this stuff is going to have to be worked out. And with any luck, they'll figure out a way to administer some of these things out of hospital. What are we going to see in the future? Well, we're going to see these drugs used beyond blood cancers. So I showed you all the ways that they're being used now in multiple myeloma and follicular lymphoma, etc. Well, they're testing them, investigating them now in solid tumors. So things like lung and prostate. I don't have the types of cancers here, I'm sorry, but I do know that bites are being investigated in prostate, and there are other CAR T therapies that have been modified in such a way to try to improve their use in solid tumors. I think there have been investigations in solid tumors with the traditional CAR T model, and they were not very impressive results. But of course, scientists are trying to figure out how to improve that. There also may be problems with, as I mentioned, CAR T therapies. They have to take a guess as to how to train that T cell. They insert a gene to help it understand how to recognize your tumor. But for people who've learned about cancer, tumor is not one bad cell that is multiplied. I mean, I guess the traditional model is one bad cell that has created clones of itself. It doesn't work that way. It's not one type of weed that takes over your garden. It's one type of weed that takes over your garden and mutates into other kinds of weeds. So sometimes you have a tumor that has many different types of cells inside of it, what we call tumor heterogeneity. And so if you train the CAR T cell to attack the one type of cell that you manage to isolate, you're not training it to kill the other types of cells. So they're working on ways to maybe deal with that. Obviously, with the bites, they don't have to worry about that thing. It's not so personalized. The bites will bind T cells regardless. And then there's this issue of, well, could we like combine these with other treatments? And right now they've been studied in isolation, but maybe they'll actually add to the effectiveness of other treatments. They could maybe even potentially reduce side effects from other treatments. So that concludes it. I'm very sorry. I'm going to provide these slides with a QR code for you to access in the future. And I do apologize for the delay, but I hope that is helpful, and I'm certainly happy to take any questions.